All right, time to go teach. Hello, and welcome to this next episode of Tutors, continuing our endomembrane system, continuing on from our last episode dealing with transcription to now go and look at translation, the process where the cell is going to use its genetic material to finally produce a polypeptide chain, which, if that polypeptide chain functions, is called a protein. And if it doesn't function, it will then have to have some other processes that occur to then make it functional and become a protein. So, targets of the day. First target is to define translation, to actually look at what translation is. Second is to identify the main structures that are going to be involved in the process of translation. And lastly, to be able to translate a section of mRNA. So if you are given a section of mRNA strand, you should be able to translate, or you should be able to determine the sequence of amino acids that it would produce. So looking at all this, translation, what is it for? Well, it's synthesizing a polypeptide from an RNA template, specifically an mRNA template. And it uses a few different things. One, it uses your mRNA. That would be your template. Next, it uses ribosomes. Ribosomes, we should remember from back in the day that they are protein and RNA, specifically rRNA, or ribosomal RNA. And this is our tRNA. And our tRNA is transfer RNA, and it carries an amino acid along with it. So we have transfer RNA, messenger RNA, and ribosomal RNA all being used in the process of translation. To build our polypeptide, the ribosome is going to move along the mRNA sequence in the 5' to 3' direction. The direction matters a lot right here. The mRNA sequence contains these things called codons. And a codon is a set of 3 nucleotides that are going to signify a specific amino acid that should be added. The tRNA, it carries that appropriate amino acid to the mRNA ribosomal complex. And how does the appropriate tRNA show up with the appropriate amino acid? Well, the tRNA, it has an anticodon. And the anticodon is the complement to the codon. So if the codon for the mRNA is an A and an A and an A, then the complement or the anticodon would be U, U, U. That is how the appropriate amino acid is added, because the tRNA has the appropriate sequence which will relate to the codon. And then, inside the ribosome, a peptide bond is formed between our different amino acids, making our polypeptide. So let's look at how this process actually takes place. First off, here is our mRNA strand, and you can see that this end over here is the 5' end, and over here off the screen would be the 3' end. So we start off with the small ribosomal subunit is going to interact with our mRNA along with a tRNA. And this tRNA is a very specific tRNA. You'll notice it's carrying the amino acid methionine. And the reason for this is that our start is always going to code for methionine. That is our start. And the codon for methionine is AUG, going from the 5' to the 3' direction. So when we look at this, going from the 5' side to the 3' side, we find AUG. That is where our start will be. So when they come in contact with that appropriate codon, and you can see the anticodon, UAC, is able to base pair with our mRNA. At this moment, the large ribosomal subunit is going to now interact. And we're going to cut away this area so we can see on the inside. And I'm not going to show the anticodon anymore. I'm going to remove the anticodon because it honestly makes it a lot harder to animate. So we're going to have it go away, and we're just going to have our tRNA now in the center, shrink everything down so we can fit this in. The ribosome is going to move toward the 3' end. But before it does that, we have a codon right there that's free within the ribosome. So a tRNA is going to bring in the appropriate amino acid. This codon of CGC, it codes for the amino acid arginine. The methionine will leave its tRNA and bond to the arginine. Now we have formed our peptide bond between those two different amino acids. The free tRNA, it will exit the ribosome, and the ribosome will shift along the mRNA one frame, or one codon, a set of 3 nucleotides. Now, our site is open again, and a new tRNA will enter carrying the appropriate amino acid. Again, we have the codon AUG, and so now it brought methionine again, because AUG always codes for methionine. Each unique codon codes for the same amino acid every single time. So now those two amino acids are bonded to the methionine. We are forming our polypeptide chain. The free tRNA exits and the ribosome shifts, and it continues to move down the mRNA strand. A new tRNA comes in. Peptide bond is formed, and the whole thing shifts. New tRNA comes in, peptide bond is formed, and we just shift until we get to a specific sequence which causes termination, when a termination factor will enter into the actual site with our ribosome. This will then cause our ribosome to break down, and then our mRNA strand will also be broken down because it is no longer needed. And all of those nucleotides will end up being recycled. They will go back into the nucleus to form or to be used in transcription again. And at the end, our end product is our polypeptide chain. We have, in this case, methionine, arginine, methionine, histidine, alanine, cysteine, and asparagine. That is our polypeptide chain. Codons are very, very important then. And there are 64 different codons, but there are only 20 different amino acids, and we have the termination, so there's only 21 different things that they code for. Some codons code for the exact same amino acid, and every one of these codons has been charted out. So if you look online, if you go onto Google and you just type codon chart, you can very quickly find 2 different main ways of determining, based upon the codon, what amino acid will be brought in. The first one that we are going to show here, this is a typical codon chart that is shown in a table format. You have one side is for the first nucleotide, the top is for the second nucleotide, and the far side is for the third nucleotide. And it's just where these different lines line up. So if we were looking at AUG, A, adenine, we would go straight across. U, we go straight down, and from G, we go right across. And where all 3 of these meet, that shows us what amino acid would be coded for. And in this case, AUG codes for methionine. Let's do a different one. Here, we have the code for CAC. That would code for histidine. And we can see it is where all 3 lines cross. And lastly, do this one more time for this. If we did UGA, that actually codes for a termination. That would code for the ribosome releasing the polypeptide strand and leaving the mRNA. The other type of codon chart looks like this. It is a pi codon chart. And I'm going to use the exact same different codons, but I'm going to show you how you read this chart. You read it from the center out. So AUG would be, starting from the center, A, and then U, and then G, and you can see again that that still codes for methionine. It's the exact same information, it's just laid out in a slightly different way. Remember the other one was C, A, C, and that coded for histidine. Again, just starting from the center and working our way out. And lastly, the other one being our termination, that was U, G, A. And that is our termination sequence, which is another way that we say the stop sequence. Termination sequence, stop sequence, it's the exact same thing. So now, why don't you give this a shot? Why don't you, using a codon chart, try to translate the following mRNA sequences? We have two, and just a hint, pay attention to the direction of each mRNA strand. Remember, this one you go that way, and this gives you these different codons, AUG, CCA, UUG, GCA, and UAG, and that will code for methionine, proline, leucine, and alanine. On the other hand, the bottom strand here, that one, remember, we had to read in the 5' to 3' direction, and that will give us these as our codons. AUG, CAC, CGA, UAC, GGU, and UAA. And that will code for methionine, histidine, arginine, tyrosine, and glycine. And you could have written this in the exact opposite order if you wanted to. That would be fine. That's not a problem. But just remember, you have to start from the 5' end, and AUG, that is your start. You don't start right where the mRNA nucleotides begin. The start codon is AUG, and it always codes for methionine. So in summary, translation, that is polypeptide synthesis. That's where you're actually putting together the amino acids to build your polypeptide and possibly building your proteins. The mRNA, that part is going to carry the codons from the nucleus, the instructions from the nucleus out into the cytoplasm. And that is going to determine the primary structure of your polypeptide chain. This goes back to a few lessons ago where we talked about the different structures of the polypeptide chain, and the primary structure is the sequence of those amino acids, the order of amino acids, and that's determined by the codons on the mRNA. The tRNA complex contains both the anticodon, so it can interact with the mRNA strand appropriately, as well as the correct amino acid, so you build the appropriate polypeptide. And it's all put together by the ribosome as the ribosome moves along that mRNA strand, taking those amino acids and forming the peptide bond between each of those amino acids that were brought to it by the tRNA. And that is polypeptide synthesis, or translation. And that's it for this time. Be awesome, stay awesome.